Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be explaining you all about ribonucleotide reductase enzyme function and how it is going to help in the formation of DNA, that is deoxyribonucleic acid. Let's get into the details. Now, the conversion of ribonucleotides into deoxyribonucleotides, it needs a ribonucleotide reductase enzyme. So, basically here what happens, the nucleotide diphosphates, which I am writing it as NDPs, all nucleotide diphosphates, will be converted into deoxynucleotide diphosphate. Basically, the ribose sugar here in a nucleotide diphosphate will be converted into deoxyribose sugar. That's the function of ribonucleotide reductase. What are the examples for nucleotide diphosphates? Nucleotide diphosphates are adenosine diphosphate, guanosine diphosphate, cytidine diphosphate, uridine diphosphate. These are all nucleotide diphosphates which will be converted into their corresponding deoxy form like deoxyadenosine diphosphate, deoxyguanosine diphosphate, deoxycytidine diphosphate and deoxyuridine diphosphate. So these are the products of the reaction. So that's what I have written here. So ribonucleotide reductase enzyme complex, the substrate for them is nucleotide diphosphates, all four of them, ADP, GDP, CDP and UDP, and the products of this particular enzyme reaction is deoxynucleotide diphosphates, that is dADP, dGDP, dCDP and dUDP. Now the donor of reducing equivalents in this particular reaction is thioredoxin, and the NADPH plus is necessary for maintaining thioredoxin in its reduced state. So with the help of all these things, that is with the availability of substrates and with the availability of reduced thioredoxin and sufficient availability of NADPH plus, which is predominantly coming from pentose phosphate pathway. So ribonucleotide reductase is able to convert all nucleotide diphosphates into their deoxynucleotide diphosphates. Let's look into the reaction proper here. So here is the base and the sugar. Sugar is a ribose. This is a ribose sugar that is attached to the base and it has two phosphates there. That's why general name nucleotide diphosphate, depending on what is the base that is attached here. So it will change. It can be adenosine diphosphate, guanosine diphosphate, cytidine diphosphate, uridine diphosphate. So different types of diphosphate forms can be present there. Now thioredoxin, which has to be in the form of reduced state, SH and SH should be available for a thioredoxin to participate in ribonucleotide reductase catalyzed reaction, because what happens in the ribonucleotide reductase reaction is the 3' hydroxyl group which has OH here, and that OH is taken out and it will be added with these two protons to release H2O, water molecule will be released. Okay, 2H from SH and SH, thioredoxin gives those two protons, and oxygen will come from this hydroxyl group, and that's where the 3' hydroxyl group is now dehydroxylated, and that's a deoxyribose. So that is D, and then nucleotide diphosphate, deoxyribonucleotide diphosphate. Okay, in this process thioredoxin is oxidized, and this oxidized thioredoxin need to be reduced back into its reduced form. For that NADPH plus is necessary, and that reaction is catalyzed by thioredoxin reductase enzyme. And overall, with the help of ribonucleotide reductase and thioredoxin reductase, NDP will be converted into dNDP molecules. So that means ADP is converted to dADP, GDP is converted to dGDP, CDP is converted to dGDP, dCDP, and UDP is converted into dUDP. This is how deoxy forms of nucleotides are synthesized, and they will eventually go towards DNA biosynthesis. So that means ribonucleotide reductase is the most important enzyme in the formation of DNA. Okay, so this most important enzyme, it is obviously the complex enzyme of all, and the regulation is complex process. So it has two important sites here. One is activity site or a regulatory site, other is substrate binding site or an active site. So at the active site, nucleotide diphosphates will go and bind, so that they will be converted into deoxynucleotide diphosphates. Whereas at the activity site or regulatory site, so regulatory molecules will go and bind. So like ATP is a regulatory molecule, dADP is a regulatory molecule. So all these regulatory molecules will go and bind to regulatory site and modulate the activity of the enzyme. Okay, so ATP bound to the activity site or the regulatory site, it is going to activate ribonucleotide reductase enzyme. Whereas a dADP binding to the regulatory site, it's going to inhibit ribonucleotide reductase enzyme. So ATP is an activator of ribonucleotide reductase, dADP is an inhibitor of ribonucleotide reductase. Now let's get into more details on the regulation of ribonucleotide reductase. So here is the active site, A for active site there, that is the substrate binding site, and R for regulatory site on this side. Okay, so whenever ATP binds to the activity site, that's the regulatory site, ATP binds here. So at that time, so UDP and CDPs will come and bind to the active site of an enzyme, that is the substrate binding site. So CDP is converted into dCDP, dCDP into dCDP, and UDP is converted into dUDP, dUDP into dUMP, and dUMP into dTMP, and that dTMP eventually converted into dTTP, and this dTTP will go and bind to the regulatory site and replace this ATP from the regulatory site, and dTTP binds there. And when the dTTP binds to the regulatory site, at that time GDP will come and bind to the active site here. And this GDP is converted into dGDP, which is converted into dGDP, and that dGDP will come and replace this dTTP and bind to the regulatory site. dGDP bound to regulatory site now. When the dGDP bound to the regulatory site, at that time ADP comes in here. And this ADP will be converted into dADP. dADP is converted to dATP. When there is sufficient dATP, this will come and bind to regulatory site, and it's going to inhibit or decrease the activity of ribonucleotide reductase enzyme. That means by the time dATPs are elevated in the cytoplasm, by that time already other deoxynucleotide triphosphates forms are elevated. That means dTTPs are sufficiently available, dCTPs are sufficiently available, and also dGDPs are sufficiently available. Then only there will be increase in dATP, which will go and bind to regulatory site and act as a negative modulator on ribonucleotide reductase enzyme. So this is a little bit complicated regulatory mechanism that you must be aware of when it comes to ribonucleotide reductase enzyme. So simple point to remember is ATP is positive modulator and dATP is a negative modulator. How that happens? It is shown here in this particular slide where I have written all of that. Coming to ribonucleotide reductase inhibitors. So we have methoxylene gadolinium, which is an inhibitor of thioredoxin reductase and also ribonucleotide reductase. Thereby, this can be used in treatment of brain tumor. Hydroxyurea. It inhibits ribonucleotide reductase, and it is used as in a treatment of myeloproliferative disorders. And one of the side effect of hydroxyurea is it increases the gamma chain synthesis. And this increase in gamma chain synthesis leading to elevation of fetal hemoglobin. It is this particular effect is used in the treatment of sickle cell anemia, thereby decrease the sickle cell crisis. Gemcitabine is a nucleoside analog and it is irreversibly inhibiting ribonucleotide reductase enzyme. So these are some of the drugs that are inhibiting ribonucleotide reductase enzyme. So that's all about ribonucleotide reductase complex. Thank you.